Morning, everybody. Hope you're enjoying your spring break. I'm thinking about anatomy and physiology on my break. And I wanted to help you guys because the lab practical, you had a few questions that weren't strictly about anatomy because we've also worked with the concept of diffusion and things moving around in lab. So you had some questions on the lab practical about whether or not things would move, basically. So I want to help you think through that because the difference between most of you guys and me is that I have a set mental program that my brain runs whenever I'm being asked a question about whether or not molecules or atoms are going to go somewhere, whether or not they're going to shift their positions from where they currently are to someplace else. And when you get to that point, when you're able to run that mental program kind of as a chunk in your brain, becomes automatic, you can apply that same logic to any new situation and any new collection of molecules and atoms, whether you've seen them before or not, which feels like a superpower because you don't have to memorize everything. So what I want to do is I'm going to walk you through, I'm going to externalize my mental thought process on that, excuse me, and I'm going to really make clear about first I think about this, next I think about that. And you may think through these things in a slightly different order as you get used to it, but if you want to borrow my method, absolutely borrow it. Because, again, that's the only difference between us. I've had a lot of practice applying these steps in my brain, so I don't really even have to think about them that hard, and that frees up part of my brain to think about kind of the new aspects of the problem, because I'm not constantly going back over okay, what do I have to ask first? All right, so I'm going to show you this scenario that I drew up. And this is a very common scenario in physiology, and there's a lot going on, but once you have that mental program, you can think through it pretty quickly. All right, so in this image, I got a bunch of stuff, and we got to remember that, everything in physiology is animated because all these molecules are constantly in motion. They're smacking into each other. Some of them are attracted to each other. Some of them are repelled by each other. So it's hard to show in a 2D image, but we know they're all bouncing around constantly, all right? You guys have done some simulations with diffusion because what we're asking with diffusion is this particular molecule I'm interested in, is it going to move in a particular direction? Now, you guys know at this point, because you've done a really good job internalizing this idea, that these kinds of movements are determined by just random motion of these particles, right? So everything's constantly moving, constantly bumping into other things. And so what we're interested in is there going to be an overall direction to that movement that we're going to see? Or are we not going to see that? And remember that diffusion specifically is saying, if we're going to simplify it to a point where my 9-year-old can understand it, diffusion just means things spread out. So if things start out crowded in one location and they're less crowded in another location, if you give them time just by that random motion, they will spread out until they're equal everywhere. They're still going to be moving, but you won't see any overall spread in a particular direction. So that's what we're asking here. We're asking, okay, of these molecules, do any of them have that tendency? Are they going to diffuse? Are they currently equal everywhere? And if they're not currently equal everywhere, then how do I predict that their movement is going to get them to that point where they are equal everywhere? So the terms we're using for that is if they're not already at equilibrium, so if they're not already equally spread out everywhere, then what we're saying is there's a concentration gradient. A concentration gradient is saying there's more of them in one place than in another. So if you just leave them alone, when that's the situation, they will spread until they're equal everywhere. So what we want to look at in this particular scenario is that the case? And to do that, you have to look one molecule type at a time, all right? So let's take, for instance, so this setup these are very common things we have in physiology. So let's look at our potassium ions first, okay? So we've got our K pluses. Those are potassium ions. And the first thing I ask myself is, okay, I'm being asked about diffusion. I'm being asked if I leave this situation alone and just let these things bounce around the way they do randomly, when I come back later, how will the picture look different? All right, so when I look at my picture, I've got a potassium ion there and there, and then I've got them here, 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 and here. Okay, so having identified, that's my first step. I say, okay, basically, is there a concentration gradient? Are they more crowded in one area than another? Because if there's a concentration gradient and I leave them alone, I don't have to do anything to make them spread out. I don't have to force them to do that. I don't have to push them. I don't have to pull them. They're just going to, through random motion, they're going to spread until they're equal everywhere. So when I look at potassium, I can see that I've got more potassiums over here in this corner of my image. And so I should predict that if I leave them alone and don't do anything to them, I'm going to expect that, some of these like potassiums, and they're random. They may like take kind of crazy paths. But eventually what we're going to have is those potassiums are going to spread out until they're equal everywhere in the picture. So I'm going to note down that. So I'm just going to run down my list. I'm going to say, okay, for each kind of atom or molecule in this picture, is there a concentration gradient currently? And if there is, how would I predict the movement will be? So for potassium, I'm going to predict overall there's going to be a movement of potassiums in that direction. And then when the whole process is done, I'll expect to see potassiums equally spread out around this whole picture. They'll be evenly spread. They'll be at equilibrium everywhere in the picture. All right, so that's my situation with potassium. So for potassium, I'm just going to make a little note. Potassium, K plus, is going to move overall in that direction. Right, so that's my little notation. I've thought through that one. So that's my first question, and I've done it for potassium. All right, so now I'm going to do it for sodium. All right, so sodium is our Na plus ions right here, these guys. So when I look at them, I've got this 1, 2, 3, 4, 5, 6, 7, 8, 9 over here on the right upper corner, and I've only got 2 over here. So that means I've established there is a concentration gradient. They are more crowded in one location than another. If I just leave them alone, I don't have to work to make them spread out. They're just going to bounce around, and eventually they're going to be equal everywhere. So that means I'm going to say for sodium, I'm going to write my little notation saying, okay, if I leave them alone and do nothing, sodium would tend to spread in the other direction until it's equal everywhere in the picture. So I'm going to do that for each thing, right? So let's see, for oxygen, I've got my oxygens here. So when I count them up, I've got 1, 2, 3, 4, 5, 6, 7 oxygens on the upper right. And let's see down here. Actually, I have no oxygens down here in this area. So that's a big concentration gradient. The bigger the difference, the faster you're going to see the spread. So if I have a big pileup of oxygen in one area, then I should expect that when I leave it alone, it's just going to spread itself equally everywhere. And that's going to happen pretty quickly because there's a big pileup in one location. So for my oxygens, I'm going to predict my oxygens are going to move in this direction. That's where their concentration gradient will lead them. And then finally, if I look at my CO2s, I've got 1 CO2 over here, and then I've got like a lot more down here. So again, that whole assessment that my brain is doing, my first step is there a pileup of this particular molecule or atom? Where is the pileup? Where is there less of them? And I know if I just leave it alone, they're just going to bounce around and smack into each other randomly until they're equal everywhere. So that means that for carbon dioxide, my overall movement is going to be this way, right? So for CO2, they're going to tend to spread up and to the right. All right, so that was my first mental step, is just looking at everything in this picture, and then molecule and atom by atom and molecule by molecule, I've figured out, is there a concentration gradient? In this particular situation, yes, there is for every single one of these things, but they're not all in the same direction. Some of them are going to spread in one direction, others are going to spread in another direction, and if I left this alone and came back in 20 minutes, I would expect everything to be equally spread out, just like a well-blended soup of these different molecules. There wouldn't be a pileup on one side or the other, because that's what diffusion tells us, is that by random motion, these things will just spread until they're at equilibrium. All right, so that's great. That's the first step. I've found my concentration gradient, step 1. Step 2, predict which way these things will spread through diffusion once I know there's a concentration gradient. Because if I had found no concentration gradient, I wouldn't predict any particular movement in a given direction. So I've done the first 2 things. All right, so then we have another issue that you guys had to deal with. In this picture, as I've drawn it, there's nothing in the way of these things moving. So you leave them alone, they're just going to mix and spread, and then they're going to end up equally everywhere. The next mental step you want to do, and this is kind of a separate step. So first, I just looked at where do I have a pileup and where do I not, predict which way they're going to move. But you guys had an example where you had a cell membrane in the way, right? So I'm just going to erase. No, I'm going to leave that for now. You had a cell membrane in the way. So these things aren't necessarily all free to mix and bounce around and come and go as they please. There may be some that can do that, but there may be others that cannot. So once I've established, are there concentration gradients in this picture? If there are, which direction would these different things tend to move? And then finally, that last question is, all right, that's where they would tend to move, but I have to figure out, can they get there? Or is there a barrier in their way? And a cell membrane is a barrier to some things, but not to others. So your first step, is there a concentration gradient? Second step, if there is, which direction do I predict the movement to be in overall? Your third step, for the particular molecule I'm interested in, can it do what I just predicted? Or is there something in its way preventing it from doing so? So we know that the middle of a cell membrane is nonpolar, it's waxy, it's fatty. So I'm just going to put all these things. It is non-magnetic. So for the reasons we've talked about before, things that are magnetic, things that are charged, things that are polar, they're not going to go into that cell membrane because they're going to be too busy being attracted to their magnetic neighbors. And you'd have to overpower that attraction to try to pull them in to the cell membrane. They're not going to do that on their own. They're not going to be, magnets aren't going to naturally draw themselves toward non-magnetic things. You don't see a magnet leaving your refrigerator and being drawn toward your wooden cabinets instead. That doesn't even make any sense because magnetic things stay with magnetic things because that's their attraction. So we then have to look at all the things in this picture and figure out, okay, which things are going to act magnetic, are either polar or are charged. And we know that those things, if the cell membrane is just a continuous cell membrane with no holes through it, those things aren't going to be able to get across. So if I look, for instance, at sodium, which I established would tend to go across the cell membrane this way, if it could, I have to figure out, well, can it? And so for sodium has a charge on it, this big-ass plus sign on it. And that's going to act as a very strong magnet. So we have to also remember that this whole environment has all this water in it, right? So every environment in the body is basically a watery environment. So I've got these sodiums that are busy being attracted to these water molecules, right? So I've got my little magnetic attractions between these two things. I'm trying to make this look like a heart. Oh, that's really deformed. All right, so those things are attracted to each other. So that means that this sodium is going to be kind of held where it is. So even though we predicted that it would come this way, it's going to come up against this cell membrane and not be able to cross it because it's too busy being drawn backwards towards the watery environment outside the cell. So this is complicated. It's another level of thinking you have to do. And until all these thoughts become automatic and just run as a program in your brain, it's going to feel like your brain is just struggling. So we've established that anything with a charge on it is going to kind of be stranded on one side of a cell membrane because it's going to hang out with the other magnetic things. It's not going to be drawn in here where it's all waxy, fatty, non-polar. There's nothing to pull it in there. Now, it's a different situation if there's a channel for sodium. So you have to think about all these things. But what you want to think about is, okay, if there's no opening for sodium, it's not going to cross a cell membrane on its own. It's going to stay where it is. So even though I predicted this movement when I ran my mental program about concentration gradients, that next level of thinking about a cell membrane being in the way changes everything because that sodium is kind of stuck where it is. So sodium, I don't predict is actually going to cross this cell membrane. Now, if you tell me that you've made channels for sodium and it has a way across and the channels are open, all right, then that changes my thinking. But just a cell membrane with no information about sodium channels being open, no, sodium's going to stay where it is. Even though it has a concentration gradient, even though I predicted that diffusion would make it spread down and to the left, it can't go because there's a wall in front of it. All right, so let's take a different example, right? Let's look at our carbon dioxides. All right, so carbon dioxide right here, we figure they're going to go this way. All right, so we get asked the same question. I predicted that they would diffuse up and to the right, but now I've introduced this extra thing where there is a cell membrane in its way. So I have to figure, is the cell membrane going to hold it back or is it going to cruise across? Now, anything that's non-magnetic, anything that's non-polar is going to easily cruise across a cell membrane because if it's non-magnetic, then it's not going to be held in place. So we've got all of our water molecules here. The whole background is water molecules, and they all act like little magnets. So if those water molecules are kind of holding whatever we're interested in place by its attractions to it, then that thing is not going to drift across the cell membrane. But if there's nothing holding it, if it's not magnetic, if it's not polar, then it'll just kind of drift along its concentration gradient. So when we look at carbon dioxide, and this goes back to like, how do we know whether something's polar? So that's something for, if you don't have that down yet, that's another mental program you need to get good at running. But I know that carbon dioxide is a non-polar molecule. So because of that, it's not magnetic. It's not being held in place by its attractions to water because it has no magnetism. So it's free to just move down its concentration gradient, and it's going to cruise right across this cell membrane because it basically doesn't see that cell membrane as a barrier. So it's free to move down its concentration gradient. So I would expect, so sodium's stuck in place, can't move down its concentration gradient, but CO2 can move. This picture is getting very crowded. CO2 can move. So that's kind of that's the mental program you want to do. Number one, is there a concentration gradient for this particular thing? Number two, if there is a concentration gradient, which direction do I expect it to spread out in just on its own without me doing anything? Step three, once I've predicted that, I have to figure out, is it free to move the way I predicted that it would? So you have to ask yourself, okay, is there a cell membrane in the way? Does that particular molecule cross cell membranes by itself? Or does it need a channel? And then we're going to get into this issue of, well, which kinds of channels does a cell membrane have? Which ones are open at any given time? What I want you to always be assuming is, the one thing we know about cell membranes is there's always water channels. There's always those aquaporins, and aquaporins are always open. So when you do these mental predictions, one thing you can be certain of is when I'm looking at a cell membrane, there's always going to be channels open for water. Any other kind of channel, I'm going to have to ask myself, is the channel there and is it open? Because if you look at something like sodium, without an open channel, sodium's stuck on whichever side of the membrane it's currently on. If you make a channel for sodium and you open that channel, then in this picture, we would expect that sodium would come into the cell, would cross the cell in the direction that we originally predicted. So know this is a lot. You guys are coming along. You are making progress. Once this becomes an automatic set of steps in your brain, you will be liberated because you won't be using so much bandwidth mentally to think through these steps. These steps will become automatic. Then you get to shift your attention to a new problem. And this stuff becomes easy. Just like when you learned how to ride a bike or drive a car or do anything that's now automatic to you, it wasn't always automatic. You used to have to think about each step separately. And that overwhelms your brain, so you can't think about anything else. When you got to that point where you knew how to do that thing, your brain was free to think about new things, to add details, to get better at it. This works the same way. You absolutely can learn this. So I'm going to ask you to take this same picture and run through that same mental program for the two that I didn't cover. So we've got oxygen molecules in this picture, and we've got potassium ions in this picture. And I've actually drawn this the way human cells are literally set up most of the time. So if you can predict this, you can predict a lot of physiology, and you become superpowered. So try that mental model. I can give you lots more practice with this, and that's the only way to get these things. So good luck, think through it. We're going to see lots more of this stuff. You're going to see it in lots of different contexts. I believe that you can do this. I believe that you can get there. You absolutely can learn this. So get to work, and I'll see you guys next time.